Hi everyone, my name is Hsu and I'm an assistant professor of chemistry and pharmacology at the University of Virginia. My laboratory is interested in studying how nature controls cell signaling through lipid metabolism. I'm here today with my graduate student Caroline Franks, who will describe our exciting new findings using chemical proteomics and quantitative mass spectrometry to identify regions of lipid kinase active sites important for substrate and inhibitor recognition. She is a leading author on our manuscript, and I will let her describe our findings in detail.
All right, thank you. So my recent work focuses on the lipid kinase DGK, or diacylglycerol kinase, and these enzymes are responsible for catalyzing the conversion of diacylglycerol, or DAG, to phosphatidic acid, or PA, by phosphorylation. There are 10 mammalian isoforms of DGK, containing in common 2 or 3 characteristic C1 domains and the catalytic region. To date, the field has been puzzled by which domains on DGKs are responsible for catalytic versus non-catalytic functions. So in order to address this question, we have developed a novel assay that permits quantitation of active DGK proteins at the peptide level. The assay utilizes an ATP probe that detects recombinant and endogenous kinase activity by interacting with the ATP binding pocket to form a covalent adduct with a conserved lysine to tag active kinases with a disthiobiotin tag, which allows for detection and enrichment. We can use this assay along with mass spectrometry to identify probe-modified peptides that are sensitive to competition with substrates such as ATP, as well as inhibitors. These peptides represent protein regions of DGKs responsible for binding to small molecules in their native states. We first used this assay to define the ATP binding pocket of DGKs. From these proteomic studies, we identified a DGK-specific ATP binding motif that's different than protein kinases. These molecular details are important for understanding why DGKs have a preference for lipid substrates and how to design drug-like molecules to target these enzymes. Next, we evaluated the selectivity of a serotonin receptor antagonist called ritanserin against DGK alpha. Ritanserin possesses high structural similarity to established DGK alpha inhibitors and was shown to be safe in human trials, providing a potential opportunity for rapid translation of our findings via drug repurposing. When we compared the inhibition profiles of ATP versus ritanserin, we discovered these small molecules bind to the DGK alpha active site at overlapping and distinct binding sites. The first C1 domain was a primary binding site mediating ritanserin inhibition of DGK alpha. This finding was important because the role of C1 domains in DGKs is still poorly understood. Importantly, ritanserin was inactive against any other DGK subtypes tested, suggesting that the binding mode of this inhibitor can guide development of isoform selective inhibitors. We are currently exploring fragments of ritanserin to test our hypothesis with the goal of developing selective inhibitors for each mammalian DGK isoform. So that's all we have today. Thanks for watching, and be sure to check out our paper, The Ligand Binding Landscape of Diacylglycerol Kinases, available now on Cell Chemical Biology.